like in a movie or something like this. And it's a human-sized problem. It's not an uncaring world with scary things that we don't know about in it, which I think is more accurate. And so it's like a human tendency to want to make this small, human size, when I don't think the problem really is. And I don't think that'll help us deal with the next SARS and the next one and the next one. Yeah, eventually we'll figure this out. I don't know if that's a good answer or not. Yeah.
I guess so. So would you agree if I say that from the evolutionary microbiology point of view, we could say that even if it is not direct to the senses of the people, it's easier having this type of virus from natural selection than from designing in the lab. That's the point, as that maybe where Occam's razor's thinking is going to. So it's even easier if you do the probabilistic calculation of all of that. It's easier having it from natural selection, even if that is not very obvious to the general audience.
Yeah, I think the p-value on the calculation that it came from people is so small that you cannot see it. Yeah, essentially, yes.
Okay, thank you so much. Yeah. Mark, do you want to come back?
Yeah, I think one follow-up from that is I agree. It seems really clear this is a natural virus. What do you think about the idea that rather than a spillover, it escaped from the lab, right? So this is the latest version of the conspiracy, that it's either escaped or been released from a lab, and if you, depending on which side of the ocean you're on, you might accuse one side or the other of releasing it.
I think when we talk about this, it tends to come up in the newspaper as intelligence sources said this. And I always wonder how these intelligence sources, they keep forgetting that everything they know is top secret and they're not supposed to tell anybody. So embarrassing. It looks like propaganda from both sides, honestly. I haven't seen any scientific evidence at all. And I think what it amounts to is like if somebody says, look, behind you, right over there. Oh my goodness, like that. Yeah, so that you'll look behind you for a minute. I don't think there's any substance to it. I think it is a thing that people talk about and worry about, but it is not real.
Okay. So, Camilo, I would like to translate a question from the chat. And it's related with domestic animals. So whether this virus can infect domestic animals. And also that we have seen these reports in the news that a tiger in a New York zoo was infected. So is this true? It can be possible.
Yeah, there's a paper that came out of, I think it was out of China. And they actually tried to experimentally infect lots of different kinds of farm and domestic animals. And they found that cats could get it and held on to it. They had a nice little course of the disease where it went up and back down. They didn't show there was any proper disease. The cats didn't seem to be sick, but they did have the virus for a while. And so it would appear in several cases now, in Hong Kong, I know they've found this several times, that if a person in the house has the virus, they can give it to the cat. We have not yet seen a case where a cat has definitely given it to a person. I don't think that's something we need to worry about. In that same study, there was a dog they were able to give the virus to for just a little while, and I think there's been one case where they found it in the dog, and it was enteric, so it was in the intestines. And so that's not particularly likely to spread. We still haven't been able to culture any viruses from human intestines. Those digestive enzymes just beat up the virus a lot. So you can detect it there. It's the best place to detect it. But it's probably not an enormous risk factor. But in cats, if I remember right, it was in the lungs. And so potentially, this could be a possibility. And I think they looked at other animals, but I don't think there is good evidence for other kinds of animals. But I would be watching other... So cats and dogs are not so far apart from each other on a phylogenetic tree. And so something like bears might be susceptible. But we don't usually have them in our homes, so I don't think we need to worry. Or if you have a bear in your home, maybe you have something more to worry about than the coronavirus. How's that? Yeah.
So, Ben, at this point, what do you think about the evolutionary trajectory of the virus? So I know that there is now quite a lot of interesting information about it, with all the sequences that are being kept from all around the world. What do you think about the trajectory of the virus? Should we be so worried about it? Is it bringing us bad news, good news, or nothing to take a position about it?
Yeah, okay, a mixture of all these things. How's that? For a non-answer. Yeah. Okay, so let's do good news first. Build people up and then beat them down. That's a good way to do it. So the good news is that this virus has, at least in, I believe it is Singapore, picked up, Singapore, Taiwan, I don't know, somewhere over there. It's picked up a big mutation where it's actually got a deletion that knocked out 1.5 genes. In regular SARS coronavirus, these genes actually made the virus grow better in human cells. And so knocking out those genes, which is something the virus has done, not someone has done, would potentially make the virus a little less likely to spread. And so that's probably a good thing. There was another report out of China where they took some of the mutations in the spike protein and made those and then tested them out. And they tested them out in a kind of a monkey cell called the Vero cell. It's very easy to work with. I love them, but it's not a human immune system. It's not the full experience of the virus. But they did find some differences. There were some that were, I think the highest reported was about 270-fold better at entering cells. Not giantly different. With viruses, we're always dealing with logs, and so this is, just a little bit better. But worth considering. And I think over the past week, what I've seen in like bioRxiv and medRxiv is modeling group after modeling group looking at all the mutations they can find and remodeling the proteins, because we now have structures for most of them, and saying, it's going to change this protein in this way. And it's going to take a while to actually confirm those analyses with biology, because biology is just slower than the computational science. But I'm real interested to see what comes out. We know the virus changes at a rate of about 30 mutations per year, which is about the same rate as influenza virus. So it's not going to be a problem in terms of vaccination any more than influenza is a problem. We still use the 2009 strain of H1N1 influenza as a vaccine, and likely an early strain of coronavirus would still probably work very well against anything that's out there in the world right now, if we can find a way to actually vaccinate that works, which we do not have yet.
Of course, from the point of view of diagnosis, so are those changes that are taking place could affect diagnosis in some way? You think?
There are not many changes historically so most of the diagnosis is based on a quantitative PCR, and they're looking for regions in the nucleocapsid or nucleoprotein gene. They look for this because the virus will make about 25 copies of nucleoprotein gene for every copy of the genome, which also has nucleoprotein gene on it. And so that's just a good, easy way to get the best possible detection. Even so, we know that diagnostics have a lower threshold. They need to have at least 1 million viruses in order to be able to detect any virus at all. Less than 1 million active viruses, and the answer is always no, which is kind of a problem. And so what we've seen around the world is a lot of people being released from hospital when their throat or their nose does not have viruses in it, but they still have virus in their intestines and their lower lungs. And so sometimes it comes back, and then people are worried that, oh no, there's a reinfection, something like this, when it was just that the infection was never stopped in the first place. I have a relative who is currently in the hospital and has been there for almost a month now, and is still virus positive. And it can hang around for a long time.
Hey, thank you. Mark, do you want to come back to the stage?
I think you've got some questions in your chat there that maybe we could get to.
Yeah, so Benjamin, there are several questions, and all of them are related with the immunity treatment of the virus, and I will try to do it one by one in the order they were formulated.
All right, let's go.
The first one is how much we know about the treatment with blood of people recovered of COVID-19.
Yeah, so this works. This is a strategy that works very well against a few viruses. For example, Junin virus causes Argentine hemorrhagic fever. And for that virus, transfusion of a little bit of plasma from someone who has recovered will have neutralizing antibodies, and that will absolutely shut down the virus. That's a virus that does not grow very well in people and where there are very abundant neutralizing antibodies. That doesn't seem to be the case for coronavirus, and it seems to be quite variable. About 1 out of 3 people in the limited data that we have so far actually doesn't make these neutralizing antibodies, which is an antibody that can stop the virus directly. And the other third is probably relying on some other component of the immune system, like T cells, to clear out the virus. So I am skeptical. My relative in the hospital once again has just got one of these transfusions. It's a thing that under international rules we can try without extra approval as long as it's done in a hospital in a controlled way. And I think you want to be careful there that people don't do it on their own, because in addition to antibodies in the blood, there are sometimes HIV and a lot of other problems in there, and so you have to do it in a controlled way. It doesn't seem to hurt, but for things like Ebola and many other viruses, it doesn't really seem to help enough to make a difference. So worth trying and easy to try, but I don't have a great deal of faith in
that. Juan is open the microphone.
Sorry. Yeah, so let's keep talking about immunity, because many of the questions are related to this feature. So if there is at least one case of immunity against the coronavirus in Europe or in the United States, and a question that is becoming more and more worrying. So is this going to become a seasonal entity? So every year we will have this coronavirus affecting us.
As I've said from the beginning, that can only happen if we really mess up. And there are some parts in the world, including perhaps the one that I'm sitting in, that seem to be doing their best, to mess up. I keep going down. I'm trying to hide from the sun, but it's not completely successful. So what we know about immunity from other human coronaviruses is that you can get multiple human coronaviruses at the same time. There's good evidence for this. And you won't get the same human coronavirus twice within a year. But after perhaps 16 months, that's when immunity seems to go down and you seem to be able to get that same coronavirus again. From the antibodies in those very limited studies, it looks like something maybe 6 months to a year is about all the protection you could expect. But it's going to be variable. There are people that have had mild COVID that just do not develop much of an immune response at all. And so this idea of, I don't know if you have this stupid idea in Colombia, but it's antibody passports. Like if you've had COVID, then they can give you a green sticker, and you can just go wherever and kiss whoever you want or whatever. Yeah, this is a very bad idea. Because it is based on total antibodies, not neutralizing antibodies, and it is not a guarantee of protection, and we know the protection is going to be quite variable based on the little information we have. So that's kind of where we are right now. We're still at a point where the models are predicting that this virus is going to go down in all the countries where it is by the end of summer. Now, these models are extrapolations based on certain things happening or staying the same, and people can be unpredictable. So with social distancing, with a stay-at-home order, we've been locked down for about 6 weeks here. It is possible, very much possible, to beat this virus. It is not that difficult even. But people are social creatures, and we struggle. We really struggle with isolation. That's what I would say.
Camilo, as there are several questions in the Zoom chat group, I don't know whether I could go on with these questions, or do you want to go for the Facebook questions?
Let's go on with the ones in Zoom first.
Okay.
So ask one more, and I will ask one of the Facebooks after that.
Okay. So there is I think a very interesting question also. If there is any evidence between the new variants of the virus and the clinical outcome. So there is an explanation for the 60,000 deaths in the United States as compared to, I don't know, 1,000 in other countries with a lot of cases also as compared to the population.
Yeah, so there are 4 very early mutations that all appear in China. The earliest branches are all back in China, and they trace back on the phylogenetic tree to somewhere in December, most likely, around mid-December in China. And we think the origin for this is also in China. The first month's worth of sequence data is all from Wuhan, China. So that's still very good scientific conclusion. The 4th strain of those, just arbitrarily numbered strains, is the one that's spread around the world. It does have a mutation in the spike gene. It is not one that is associated with particularly high amounts of disease, but there are some descendants of this that were among the ones that were tested and found to get into monkey cells a little bit better. So it is possible, but right now that is just speculative. And there's no correlation that anyone has seen yet between a particular mutation and a particular disease outcome. This virus just causes such a variable disease. I mean, it goes from nothing to death and everything in between, that it's very difficult to actually come up with a way to statistically demonstrate that any one strain does a particular thing. And also the problem is that the virus never stops moving, never stops mutating. And so you can't repeat the same experiment like you would in a laboratory out in the wild. It just, the virus keeps going. And so all we know is that we haven't got rid of it yet. And on average, the strains you would find today would be about 9 or 10 mutations different from the original strain. And probably with that S mutation and others built on top of it. There's lots of different lineages down there.
2 questions from Facebook right now. So I will explain both at the same time. One is if it is necessary to use animals always like a first line of test for vaccination, of if vaccines can be tested directly in people. And the other one is if we have to be worried about the duration of the survival of the virus on water. If in water they can survive long enough to be worried about.
Okay. Yes and no. There we go. So for animal testing, yes, in this case, it's very important. Most of the vaccine, there are many different vaccines that are being tried right now. There are 7 different ones as of last week's, the last time I checked, that are being used in people right now. And there's a list of, I think it's around 60 different vaccines that are in animal testing and scheduled to go into humans very shortly. And there are all different kinds of vaccines in there. The ones that are happening fastest are the ones that are easiest to produce. So they tend to be things like DNA vaccines or RNA vaccines. These are a great idea, the very clever idea that very seldom seems to work out in the world. Yeah, but they're a thing, again, that's worth trying. I just would like to see other approaches tried at the same time, which I think will eventually catch up on the other approaches, perhaps not so fast. As for the other half, do we have to worry about water? No, for the most part. So something like a swimming pool is going to be chlorinated, and we know that hypochlorous acid, which is going to be in those, it's going to form when those chlorine tablets go in, is very effective against the virus. It's, nothing really there. The average amount of time that a virus survives in direct sunlight is measured in minutes. The half-life was something like 3 minutes. And so, no, there's very little chance that water is going to hurt you. And it's not like someone can cough in a lake and then you know, fill up a glass and catch COVID. It seems to be still from very close, very direct human contact, the kind that we as social creatures really like. That seems to be how the virus is spreading.
Vanessa, some questions.
Yeah, in here in the chat we have a lot of questions. So there is a technical question, and it's regarding the protocol to make the diagnostics of the virus. So we are using in Colombia the Berlin protocol, and is based on the RDRP, E, and N genes. How good are those genes taking into account the genetic variation of these viruses?
Yeah, RDRP doesn't vary at all. Here, I'm going to move so I don't have to hide from the sunlight. There we go. Bad framing, but yeah, no more hiding. All right. The problem with, so the good side about RDRP is that, yeah, it's not going to change, and you can actually make pan-coronavirus primers that will amplify any known coronavirus, because there are regions of the RDRP that just are not able to change and still have a functional polymerase. So that's good. The problem is that you only find the RDRP on the longest message. So coronaviruses make they make a big full genome, and then they make lots of smaller versions. A full-length SARS coronavirus is actually going to make 9 of these. But when we have that deletion, I think it's going to go down to 8 because you lose the signal and one entire gene to do that. But even with, so something like N, the nucleoprotein is going to be present on all 8 of those species. And in the little ones are made much more commonly than the big ones. So that's better in terms of sensitivity. In terms of resistance to mutation, yeah, the polymerase, I think, is better. The antibody tests that are out there are not bad, but those are mainly going to be an epidemiological tool and a public health modeling tool. They only detect the coronavirus, it looks like about 3 weeks after you get infected, which is about 2 weeks after you go to the hospital. So they're useful after the fact, but not right now.
So should I go with another one, Camilo? So another question, and I think it's also quite important, is about how much time can be the virus in surfaces, or in our clothes, or in any kind of surface.
Yeah. And so these times are calculated in different ways. There was an early study that was done in the laboratory, and it showed some different times, and it had some different problems. Like they looked at the amount of time the virus can persist as an aerosol, but there aren't particularly good ways that a human body can produce an aerosol. We're not one of these little spray misters you might use on your plants. We just have one big mouth hole, yeah, and 2 little nose holes. And they're not very good at spraying, you know, a fine mist of particles. So it seems to be on surfaces, it's something on the order of 1 to 2 days until the last virus is gone. I think there was one test on plastic in a laboratory without UV from the sunlight. And in this case, the last virus didn't disappear until about 3 days. But the virus starts breaking down immediately. And I don't think contamination from the environment is a big factor in the spread. Because if you look at the way the virus spreads, it tends to get into one person in a family, and then boom, it hits that entire cluster in the rest of the family. And it only rarely spreads outside the family. So it's mostly just getting into one person, who knows how, and then going really rapidly to all the other ones. Really seems to be driven by close contact rather than anything else. And when I'm out at the supermarket, still even now at the supermarket, even 6 months into our stay-at-home order, I see now it's down to about half of the people that don't wear masks. A week or 2 ago, it was 3 out of 4 that did not wear masks. But even these people will sometimes see a friend and just sort of pull their mask down and say, "Oh, hi, how you doing?" and have a nice close conversation. And that's how it happens. It happens when we're not paying attention, when we let our guard down, I think. Yeah. This is my experience anyway from growing SARS coronavirus in the lab. You have to always assume the worst, and you have to always try to know where the virus is. And if you don't know for sure that the virus is not there, you have to treat whatever that situation is as if the virus is there, and you have to mitigate it. You have to do whatever you have to do to make sure that you can't get infected. And that's very difficult for regular people in the real world. That's difficult for laboratory scientists in a Category 3 lab.
Ben, yeah, a couple of questions from Facebook and one from my sister in New York.
Hey,
now that you are going to the transmission thing. So one of the questions in Facebook is asking about how dangerous are actually the droplets of sneeze in the air, for example, for public transportation. How worried should we be about food transmission by food, and in general, what are the main or the most important rules that we should follow in a place like New York, where everything is very bad right now, to avoid getting infected?
Yeah, I mean, things are bad in New York. My brother lives in Manhattan, and but they're doing a good job of social distancing, and there are places like Houston and Texas that are actually policing and forcing people to wear masks if they're out getting groceries or something. And this is a good idea. So what we know about viruses is that the amount of virus that you put in increases the chance of severe disease and increases the chance of infection. With the mouse coronavirus, there's one strain that actually will go into the lungs, kind of like SARS coronavirus. And we know that you need around 1,000 particles to be inhaled in order to reproducibly infect a mouse. It's probably something around this range to infect a person. So it's not that one or two viruses that are escaping through a mask that are going to get you. It's when you cough up, if you have a little bit of phlegm or a little droplet, those droplets can contain millions of viruses. And if one of those happens to land in your eyes, nose, or mouth, we know that there are two proteins required for the virus to enter. And both those proteins are present in cells in the cornea, so your eyes can definitely get infected. Both of them are present all along the mucous membranes, but particularly in the throat and in the lung. And they're also present in the intestines in very, very high numbers. Things like hearts only have one of the two proteins, and so far there's no evidence that hearts are actually infected. But yeah, it seems that that droplet, that big droplet, is what's going to get you, and that's what a regular mask prevents. Just one of those pieces of cloth, a bandana, whatever you have, even holding your arm up like that helps some because it's going to deflect any virus coming out, and it's going to block any virus, large blobs of virus from coming in. And that's probably the key. It's kind of stupid, yeah, in comparison to all the fancy gear that we see in hospitals and stuff, but that is probably the key, and places where they do that well, this keeping distance, because the virus is going to be in like a cannonball. It goes up in the air and then it comes down and it splats on the ground. Unless you are repeatedly licking the floor around COVID patients, you are not going to get this virus from a surface. Yeah. It's going to go through the air and it's never going to hit the ground, and it's going to hit another person. That's probably how it happens, at least based on what we know so far.
Okay, what about food? In general, how
No, food is cooked, and the protein, so there's 4 proteins that make the virion, like that little spiky ball, and one of those is there's a paper that's essentially a 5-page complaint about this protein. They talk about how it unfolds and how it's just so badly behaved, and just, yeah, you can tell this person had a very bad, probably entire PhD dealing with this awful protein, but it's very temperature sensitive. These viruses are made out of the same components that you find in the human body. They're designed to work at human body temperatures and heat them up even a little bit, and they pop like popcorn. They fall apart. They're not very good. Yeah, They're not durable, rugged viruses at all. Yeah.
Okay.
So no, if your food is cooked, I think it's fine. If you have sushi and your sushi chef hates you and coughs on your food, you should consider tipping better or being a nicer person, I would say. But I don't think there's a big risk from food at all. No.
Okay, perfect. So, guys, it's 5 minutes to 11, so we were scheduled to finish at 11. I don't know. I would like to abuse from you because your time is very valuable right now, so maybe we don't have the chance to have you again. If for you or for the rest of the crowd is okay to continue a little bit longer, it will be okay because we have so many questions.
As long as people want to, it's fine. Quarantine is very boring. Yeah.
Thank you.
I don't have a lot of things to do today. That's all.
No, thank you so much. Sorry, Juanis, I wanted to give the word to Mark after this. Juanis, go ahead. What were you going to say?
Okay. So there is a question from one of Diana Di Filippo is one of the researchers in one of the groups of our university, which isolated the virus, and they are culturing it right now. And she is asking whether some studies report the presence of COVID-19 in fecal material for up to 4 months. Is there any evidence that these viral particles may be infective?
Yeah, good point. The virus is definitely present at least 2 weeks after full clinical recovery. And this has been demonstrated in every patient that I've seen. I've talked with Chinese doctors, and they are now not releasing people from the hospital until they have 2 negative fecal tests. Yeah, they are not even bothering to test the upper respiratory tract because that is not where you get most of the virus. And it disappears there before other places. So far, I have not yet seen a case where anyone was able to recover live virus from a fecal sample. But the 2 things that we know are that in occasional cases, something like 5%, this virus causes diarrhea. And so I think it's going to be time that the virus is resident in the intestines. The longer it's there, the more it'll be digested and destroyed. But if you have virus that's made and coming out fast, as in diarrhea, then I think there's a possibility. There was one case at the Amoy Gardens Hotel in Hong Kong, there we go, back with original SARS, where one person managed to infect an entire side of the hotel. And the only way they could have been infected is traveling through the toilet, both up and down, and to other people. Yeah, absolutely disgusting. And it's a very fancy hotel as well. There was also a report looking at COVID patients' bedrooms in hospitals. They set up ways to capture the virus from aerosols, and they could not find more than just the tiniest trace of virus in actual aerosols, because that's not how it probably is transmitted, over a long distance through an aerosol. But in the bathrooms, they did find very high amounts of virus. They just don't know if it's infectious. But I think this is something to watch out for. I think that is another possible way that people in the same home could end up all catching the virus. But no, it hasn't been demonstrated yet. And that's really cool that you've got an isolate of the virus and they're working with it. Yeah, hats off to you.
Okay, there are several questions also about the treatment. So, do you think that these remdesivir and all these treatment alternatives that are now being published are affecting the virus itself? It is working?
Yes and no. Yeah. Remdesivir is one of the more certain candidates. So we have a crystal structure of remdesivir in the polymerase. We have good molecular tests where they just have polymerase making RNA, and it puts in a remdesivir, and then it goes 1 base, 2 base, 3 bases after, and then the machine just stops. So we know the polymerase has a very narrow little channel, kind of like that, and it's pulling the RNA through and making the nucleotides right there. And remdesivir sticks up a little bit further than a normal nucleotide would. And so I think it's just getting snagged in the tunnel. The crystal structure actually had that nucleotide in the wrong place. I think they made their constructs before the other paper was out. And so they can't actually show it. But yeah, remdesivir works in the laboratory, but clinically everything is harder. And so far, there was a significant reduction in the amount of time it took for someone to get better if they had mild to moderate COVID. That seems certain. But the difference in patients who had severe COVID, it was not significant. And in cases like hydroxychloroquine and azithromycin, that's the antibiotic they were putting in those cases, they actually, in one study, had much higher death rates when they administered the treatment in people with severe COVID. The disease itself is an immunological disease. It's a macrophage cytokine storm, kind of similar to what the bird flu or Ebola virus does. It's just happening only in a particular part of the lungs. And most of the disease is coming from this. And so remdesivir is probably stopping the virus, but it wouldn't necessarily do anything to calm the cytokine storm. So I know they're trying combining interleukin-6 blockers, for example, with remdesivir. And I have yet to see if that's going to work out really well. And there are a lot of other of these sort of immunotherapy or combined antiviral and immunotherapy approaches. And yeah, some of these might work, but until the tests are done, yeah, we're not going to know the answer. They're good ideas, though.
What about penciclovir? Somebody's asking about that in Facebook. Penciclovir.
I don't know that one. I'm going to have to look at that one. Let's see. I know favipiravir, which has a couple names. I know the active ingredient in Pepcid is histidine H2 receptor blocker, and that's involved in immune proliferation. And I know that there are people that are trying to go after these things called inflammasomes, which are going to drive a particular side of a cytokine storm. But they're going to drive it more through, I don't know if you know interleukin beta, if you know your interleukins, interleukin 1 beta rather. And the COVID does not seem to have a very strong interleukin 1 beta component to it. And so I don't know that those are going to be successful. No, I'm going to have to look up this penciclovir. Yeah, if you want to read another question, I'll secretly Google on the side and see if I can give you a better answer. Yeah.
No, it's okay. No problem. So, Mark, I would like to give you the word before you have to leave, because I know that you have so many other things to do.
Thanks, Camilo. I think someone was asking before about the cases, the idea of the possibility of reinfection or reemergence of infections, particularly in those patients in South Korea. So I think that's a really important thing. And then I guess along the treatment lines, while I've got you on treatment, I think a lot of people are very interested in treatment. Do you know of anything that's in the pipeline that can be used very early in an infection, before people are critical and the viral load is not your biggest problem?
Yeah, we've got about 17 years worth of good papers. So I found penciclovir. It looks to be a derivative of these things like acyclovir and valacyclovir. They work well against herpes viruses, but they just look like a sloppy version of a nucleotide, and viruses go fast and they don't care what they put in. They're all about going fast. So theoretically, that could potentially work. I don't think there's any reason to believe it's going to be more effective than some of the other things. That's exactly what remdesivir does and favipiravir, and there are a couple other compounds like that. What was the rest? You had an actual question there, and I went a different way. What was the question again? Sorry.
I think the questions were, the treatment question is there something in the pipeline or something you can imagine, perhaps a more stable or better nucleotide analog, which could be used early in infection when the virus is your biggest problem, instead of late when your cytokine storm is your biggest problem?
Yeah. So what I would like to see more of, so a lot of people are trying to go, so all right, let's look at the different virus proteins and which ones are an easy target and which ones are just an impossibly stupid target. Step one, the first thing people go for is a thing called the main protease. It's called NSP5, and the problem with it, so the good thing about the main protease is it's involved all over the place. It cuts something like 14 different places in the viral polyprotein, and if you miss even one of those cuts, the virus is dead. Yeah. The problem with the main protease is that the active site is like an open book. And so it is very hard to get a molecule that will actually stick on that. You don't have a lot to hold on to. There are people that are trying these covalent inhibitors that will actually chemically bond to the active site. The risk is that you're going to have a higher rate of side effects with something like that. Because it's active and can chemically bond with things. Yeah. There are a lot of targets and a lot more potential targets than there are going to be viral targets in even the most infected person. Enzymes like the helicase, there are people that have come up with blockers for those, but the blockers just involve delivering so much bismuth ion into the cell, it replaces the zinc ions that the helicase needs to work. And I think if you're trying to dose up a human being with enormous amounts of bismuth, you're going to run into massive problems once again, because we have so many zinc binding proteins. That's just going to be, crazy, stupid, and probably deadly. The polymerase, yes. Polymerase is going to be the target of these nucleoside analogs, and that's a smart way to go about it. And now that we have polymerase structures, we just got the SARS-CoV polymerase structure is out in preprint form. Actually, no, there was one that came out in Science today. Now they can start developing things that will actually stick to different parts of that. And remember, enzymes are not just a static thing like a statue. They move and they open and shut. And if you can figure out what the range of motion would be, sort of model it as little springs, you can figure out how to block it or limit its motion. And there are very successful HIV drugs that work in this way. The one that I'm most excited about, and I haven't seen anything on yet, is going to be methyltransferase inhibitors. So this virus is what I would call, I don't know if the metaphor translates, but a blue collar virus, like one that works for itself and is like like the regular guy you could have a beer with. Some viruses are dirty thieves. This is like the influenza virus. When they want to get a cap for their messenger RNA, they just cut one off from a messenger RNA of the cell and use it. Yeah, they're total jerks like that. Coronavirus, if it wants that cap, it's going to build it. It's got 4 enzymes that it needs to build it. And 2 of those enzymes are methyltransferases. Inhibiting methyltransferases seems to be a very potent way to shut down viruses like this. And we know that the last methyltransferase in the last step is called NSP16, or the 2'O methyltransferase, is essential for the virus. It is a camouflage. If you turn this thing off or inhibit it, then the virus is visible to the immune system and the virus gets shut down immediately. And so I would very much like to see more studies looking at methyltransferase inhibitors. We haven't seen them. It has a decent active site pocket. I don't really know why. It's just not as popular maybe, or a harder model to work with. I'm not sure. But I think long-term, that's going to be a good solution. And other things like where they try to block the spike protein or the nucleoprotein or the M protein in the actual virion, I think these are not likely to work because the average virion is going to have something like 5,000 M proteins and 10,000 N proteins and hundreds of spikes on the outside. And you're just never going to get enough of the drug to the right place to be able to hit all of those. The virus is going to find a way. Yeah. And we don't want the virus to find a way. So hit it where it's weak. Hit it at the bottlenecks. That's what I'd like to see more of. Yeah, that's too long an answer to a very good question.
Hey, Camilo, so there is a question regarding the epidemiology and how to stop the spreading of this virus. And the question goes like this. If the virus can stay in the body for a long time, are those people carriers? And then what is the solution to stop spreading? Is there any other option apart from confinement to stop the spreading of the virus?
Yeah, boy, people would like to find some other option, wouldn't they? We see this everywhere in the world. People are getting a little crazy, especially the ones that are locked down. And yes, you see the Floridas of this world just, saying, whatever, let's just come out of quarantine and do this. That is almost certainly a bad idea. So I guess the nice thing about quarantine is that it works against any virus anywhere. It doesn't matter what the virus is. It's 100% effective. And it doesn't require scientific advancement, doesn't require an antiviral or a vaccine or anything. Once we have some of these other parts, there will be ways to have normal society and limit spread. But until we have those parts, it's got to be some version of social distancing. And so you have lots of countries experimenting with social distancing to different degrees. From like what I would call going full Wuhan, where you lock people inside and put some nails to hold the door shut and say, yeah, see you in a month. And other places where they say, yeah, distance yourself, but don't try too hard. Yeah. And we've seen some US states and some European countries. Yeah, hello, Sweden. Yeah, trying to do things like that. And we're seeing a variety of results, but the Financial Times has a nice little tracker for coronavirus. And like you said, Juanes, in I think your first question, there are all these deaths from coronavirus that we know about, but in every one of these countries, there's also a giant spike in mortality that happens to be at the same time as coronavirus appeared, and where we cannot yet confirm that those people died from coronavirus directly, but it looks like we are very much undercounting the total death rate, which is really bad. Yeah, people are very worried about getting too alarmed about this or making a bigger deal about this or overcounting the cases. But the question is, are we undercounting by threefold or are we undercounting by tenfold? My mathematical modeler friends tell me eightfold is probably about right. So yeah, we got one case and there are 7 more out there that we didn't know about, or 8 more. Yeah.
Well, thank you very much. So should we go on with another question, Camilo? Do you have one in there?
Well, one in Facebook and then go for Zoom, where we have most of the questions. In Facebook, somebody's asking about any kind of, I would say, cross immunity from a vaccine to tuberculosis. So I would add there that here, so I have heard some comments, especially from Ecuador, where many people have been saying that they have been having very bad death rates there because most of the people there was not vaccinated against tuberculosis. How true is that?
Yeah, right result, wrong mechanism. Yeah, they're trying that in the US, they're trying that around the world. So tuberculosis caused by a bacterium gets into the lungs, lives there forever unless you do a lot to try and clean it out. Very difficult problem. And if you have bacteria in your lungs and virus in your lungs, at least for influenza virus, we know this increases your risk of dying by something like 10,000-fold. So there's a good case to be made, and there's some anecdotal evidence that vaccinating for tuberculosis will just clear up one other potential problem in the lungs. And then your body is only dealing with coronavirus. It's not dealing with 2 things at once. Yeah. So it's still a good idea. It just doesn't, that vaccine doesn't protect against anything else. It's a very different virus, even from the original SARS coronavirus, and a vaccine for one probably would not protect against the other.
Okay, so another question. There are reports about the virus binding to other types of cells, just like neurons and immune cells. So is this a trait seen in other coronaviruses, or it is just a feature of this coronavirus?
Yeah, okay. All right, I like this question. There's a lot that's known about the mouse coronavirus in this. So in the human body, first of all, there are some very nice single cell papers that they were preprints 2 weeks ago. I have to think they're either out or about to be out, and I bet they're going to be in something like Nature. There's a lot of money behind these ones, and they look like really good papers. What they found was that even in macrophages, they can only barely detect virus. It looks like macrophages are not infected. There's no good evidence for virus spreading through the bloodstream. It seems as though the virus is only in certain tissues. Now, what we know from other coronaviruses is that, they can, in some circumstances, be more promiscuous. So there are other viruses. I think the limit here is the spike protein. Some spikes are very easy to activate. Some spikes are hard to activate. So the mouse coronavirus spike, if you look at it wrong, it will activate itself. And then as long as there is a protease present that can cut it at a particular spot, and the proteases that we know are cathepsin type E, works for some viruses, and a protease called Tempris2 works for other viruses. If one of these is present, then you will get activation, and then boom, the virus will fuse, even if there is no receptor at all. With SARS coronaviruses, they're playing it really safe. They have a lot of activation measures. So they have to be cut at a thing called a polybasic site that loosens up the top of the virus. Then they have to bind to a receptor that changes the shape of the virus, we think, or the spike. And then they have to be further activated by that TMPRSS2 cleavage. And unless you have both receptor and TMPRSS2 on the same cell, it looks like they are not infectable at the moment, which is good news for us. Yeah, I am happy about this. And it also makes it much easier to get good electron microscope pictures of the virus, because usually the spikes are the thing that falls apart and just makes all your hard work turn into garbage. Yeah. So, yeah, thank you, coronavirus, for that one thing. Yeah.
Okay.
Let's go with more questions from Zoom. Where
Okay.
do we so far we
Okay, so there is a